Good morning, students. Today we're going to be taking a look at mitosis. This graphic displays the life cycle of a cell. As you can see, the cell spends most of its time in interphase. Today we're going to be looking at mitosis, one of the shortest but most important phases in the life cycle of a cell. The first phase of mitosis is interphase. The nucleus is still enclosed by the nuclear envelope, and the nucleus contains one or more nucleoli. Two centrosomes have formed by duplication of a single centrosome. Centrosomes are the regions in animal cells that organize the microtubules of the spindle. The chromosomes were duplicated during the S phase, but they can't be seen yet because they have not been condensed. Here's what interphase looks like in an onion root tip cell. The next phase is prophase. In this phase, the chromatin fibers become more tightly coiled, condensing into discrete chromosomes observable with the light microscope. The nucleoli disappear. Each duplicated chromosome appears as two identical sister chromatids joined at their centromeres and, in some species, all along their arms by cohesions, sister chromatid cohesion. The mitotic spindle, named for its shape, begins to form. It is composed of the centrosomes and the microtubules that extend from them. The radial arrays of shorter microtubules that extend from the centrosomes are called asters. The centrosomes move away from each other, propelled partly by the lengthening microtubules between them. Here is prophase in a plant cell. Next comes chromatophase. This is when the nuclear envelope fragments. The microtubules extending from each chromosome can now invade the nuclear area. The chromosomes have become even more condensed. Each of the two chromatids of each chromosome now has a kinetochore, a specialized protein structure at the centromere. Some of the microtubules attach to the kinetochores, becoming kinetochore microtubules, which jerk the chromosomes back and forth. Non-kinetochore microtubules interact with those from the opposite pole of the spindle. Here is prometaphase in an animal cell. Next is metaphase. In metaphase, the centrosomes are now at the opposite poles of the cell. The chromosomes convene at the metaphase plate, a plane that is equidistant between the spindle's two poles. The chromosome centromeres lie at the metaphase plate. For each chromosome, the kinetochores of the sister chromatids are attached to kinetochore microtubules coming from opposite poles. This is metaphase in an animal cell. Anaphase is the shorter stage of mitosis. It begins when the cohesin proteins are cleaved. This allows the two sister chromatids of each pair to part suddenly. Each chromatid has thus become a full-fledged chromosome. The two liberated daughter chromosomes begin moving toward opposite ends of the cell as their kinetochore microtubules shorten. Because these microtubules are attached at the centromere region, the chromosomes move centromere first. The cell elongates as the kinetochore microtubules lengthen. By the end of anaphase, the two ends of the cell have equivalent and complete collections of chromosomes. Here is anaphase in an animal cell. Next is telophase. Two daughter nuclei form in the cell. Nuclear envelopes arise from the fragments of the parent cell's nuclear envelope and other portions of the endomembrane system. The nucleoli appear, and the chromosomes become less condensed. Any remaining spindle microtubules are depolymerized. Mitosis, the division of one nucleus into two genetically identical nuclei, is now complete. This is telophase in an animal cell. Telophase and cytokinesis are often paired together. The division of cytoplasm is usually well underway by late telophase, so the two daughter cells appear shortly after the end of mitosis. In animal cells, cytokinesis involves the formation of a cleavage furrow, which pinches the cell in two. This here is the cleavage furrow. And there you have it, all of the phases of mitosis.